This video is about stem cell concept, the stem cell concepts introducing organogenesis. Some introductory portion. So we have now explored the development of embryo fertilization through gastrulation. In the next chapter, we will see how the three germ layers interact with each other to begin organogenesis, the process of organ formation. Early organogenesis is a series of interactions between different parts of the embryo, and some of these interactions create privileged sites called the stem cell niches. These niches provide a milieu of extracellular matrices and paracrine factors that allow cells residing within them to remain relatively, undifferentiated. These relatively undifferentiated cells are stem cells, and their presence has become central to our vision of organogenesis and critical to the field of modern medicine. The stem cell concept. A stem cell can be defined as a relatively undifferentiated cell that, when it divides, produces one cell that retains its undifferentiated character, a second cell that can undergo one or more paths of differentiation. The stem cell has potential to renew itself at each division so that there is always a supply of stem cells, while also producing a daughter cell capable of responding to its environment by differentiating a particular manner. So this potential is not always realized. In some instances, stem cells divide symmetrically so that both daughter cells remain stem cells. In many cases, the stem cells remain in the niche while its sister leaves the niche and differentiates. In some organs, such as the gut, epidermis, and bone marrow, stem cells regularly divide to replace worn-out cells and repair damaged tissues. In other organs, such as the prostate and the heart, stem cells divide only under special physiological conditions, usually in response to stress or the need to repair the organ. Here you could see the picture of a multipotent stem cell along with the committed stem cell, progenitor cell, differentiated cells. So the stem cell concept. Stem cells have two distinct progeny at division, another stem cell and a more committed cell that can differentiate. So hematopoietic stem cell is isolated from human bone marrow from all the different blood cell types as well as producing more hematopoietic stem cells. These multipotent stem cells generate blood cells throughout our entire life. So in the mid 20th century, biologists thought that cell specification occurs in the early embryo and that after this stage there was only the growth of the existing parts. This idea is preserved in the classification of human development into embryonic stages where specification takes place before week 9 and later fetal stages that are characterized by growth. However, beginning in the 1960s, biologists studying blood cell development began to point out a remarkable phenomenon. All of the blood cells, red blood cells, erythrocytes, white blood cells, granulocytes, platelets, and even lymphocytes are constantly being produced in the bone marrow. Some billions of blood cells are destroyed by the spleen each hour, but an equal number of blood cells are generated to replace them. In an elegant series of experiments, Ernest M.C. Kult and James Tull demonstrated that there was a common generative cell, the hematopoietic blood forming stem cell, that gave rise to all the different types of blood cells. In a 1961 experiment, Till and McCullough injected bone marrow cells from a donor mouse into lethally irradiated mice of the same genetic strain. Some of the individual donor cells produced discrete nodules on the spleens of the host animal, and these nodules contained erythrocytes, granulocytes, and platelet precursor cells. Later experiments in which the donor marrow cells were irradiated to genetically mark each cell with random chromosome breaks confirmed that each of these different cell types in a nodule arose from a single cell and that there were lymphocytes present in some of these nodules. For this colony forming cell to be a true stem cell, however, it had to produce not only the differentiated blood cells but also more colony forming cells. This was found to be the case by taking the nodule derived from a single genetically marked colony forming cell and injecting cells of the nodule into another irradiated mouse. Many spleen colonies emerged, each of them having a small same chromosomal arrangement as the original colony. bone marrow, blood vessel, pericyte, natural killer cell, T lymphocyte, lymphoid progenitor cell, B lymphocyte, hematopoietic stem cell, multipotent stem cell, myeloid progenitor cell. Neutrophil, basophil, eosinophil, monocyte, macrophage, platelets, red blood cells. So blood forming hematopoietic stem cells and the bone marrow niche. The hematopoietic stem cell is located in the bone marrow and generates a second stem cell that is capable of becoming either a lymphocyte progenitor cell which divides to form the cells of the immune system or a myeloid stem cell which forms the blood cell precursor. The type of lineage that the cells take is regulated by the niche, which involves contact between the stem cell and the matrices of bone cells, paracrine factors from stromal cells, and the pericycles surrounding the blood vessel, and systemic hormones and neural signals. Mesenchymal stem cells also use the bone marrow as a niche. So blood forming hematopoietic stem cells and the bone marrow niche. The hematopoietic stem cell is located in the bone marrow and generates a second stem cell that is capable of becoming either a lymphocyte progenitor cell which divides to form the cells of immune system or a myeloid stem cell which forms a blood cell precursor. The type in niche that cell take is regulated by the niche which involves contact between the stem cells and the matrices of bone cells, paracrine factors for stromal cells and the pericytes surrounding the blood vessels, and systemic hormones and neural signals. So mesenchymal stem cells also use the bone marrow as a niche. Single bone marrow cell can form numerous different cell types, can also undergo self-renewal. This research on hematopoietic stem cells, which we will discuss later, led to the establishment of field of bone marrow transplantation. The concept that the different blood cell types were continuously generated by a hematopoietic stem cell was first proposed in 1909 by the Russian histologist Alexander Maximov. He is credited with coining the term stem cell to refer to the regenerative capacities of these cells. So Maximov had been a student of Oskar Hertwig, one of the leading German embryologists and one of the originators of the current epigenesis. So he professor of embryology and histology. Maximov's work was cut short by the Russian Revolution. He managed to flee the Soviet Union in 1922, having bribed a guard at the Finnish border with a bottle of laboratory ethanol. Eventually, he settled in the United States, where he co-authored the leading textbook of histology with his student William Bloom. You could see spleen colony forming cells, hematopoietic stem cells injected into an adult mouse will colonize the spleen and form colonies containing red blood cells, granulocytes or white blood cells, platelet precursors and lymphocytes. Shown here is the spleen of an irradiated mouse whose immune and blood precursor cells were abolished by radiation. This host mouse was then injected with bone marrow cells from a genetically identical mouse. When the host animal's spleen was removed 12 days later, colonies of donor blood cells were visible on its surface. Stem cell vocabulary. Numerous terms are used to describe stem cells and their usage has not always been consistent. However, there is beginning to be general agreement to how these terms are used. The names of the two major divisions of stem cells are based on their sources. Embryonic stem cells are derived from the inner cell mass of mammalian blastocysts or from fetal gamete progenitor germ cells. These cells are capable of producing all the cells of the embryo. Adult stem cells are found in the tissues of organs after the organ has matured. These stem cells, which are usually involved in replacing and repairing tissues of that particular organ, can form only a subset of cell types. Stem cell patency, the ability of a particular stem cell to generate numerous different types of differentiated cells, is its patency. And in mammals, totipotent cells are capable of forming every cell in the embryo and in addition a trophoblast cell of the placenta. The only totipotent cells are the zygote and probably the first 4 to 8 blastomeres to form prior to compaction. So potential totipotent cell zygote, source zygote, pluripotent embryonic stem cell, blastocyst, inner cell mass, multipotent stem cell, embryo, adult brain, limited differentiation potential, neuronal progenitor, brain or spinal cord, then limited division potential, differentiating neuronal precursors, beginning regions of the brain, and functional non-amniotic neuron, differentiated cells, specific areas of the brain, example of the maturational series of stem cells here applied to the differentiation of neurons. So pluripotent stem cells have the ability to become all the cell types of the embryo except trophoblast. Usually these embryonic stem cells are derived from the inner cell mass of the mammalian blastocyst. However, germ cells and germ cell tumors such as teratocarcinomas can also form pluripotent stem cells. So we were talking about pluripotent stem cells that have the ability to become all the cell types of the embryo except trophoblast. Usually these embryonic stem cells are derived from the inner cell mass of the mammalian blastocyst. However, germ cells and germ cell tumors such as teratocarcinomas can also form pluripotent stem cells. And multipotent stem cells are stem cells whose commitment is limited to a relatively small subset of all the possible cells of the body. These are usually adult stem cells. Multipotent stem cells are stem cells whose commitment is limited to a relatively small subset of all the possible cells of the body. These are usually adult stem cells. The hematopoietic stem cell, for instance, can form the granulocyte, platelet, and red blood cell lineages. Similarly, the mammary stem cell can form all the different cell types of the mammary gland. Some adult stem cells are unipotent stem cells, which are found in particular tissues and are involved in regenerating a particular type of cell. Then, spermatogonia, for example, are stem cells that give rise only to sperm. Whereas pluripotent stem cells can produce cells of the three germ layers as well as producing germ cells. The multipotent and unipotent stem cells are often grouped together as committed stem cells since they have the potential to become relatively few cell types. Then progenitor cells, although they are related to stem cells, progenitor cells are not capable of unlimited self-renewal. They have the capacity to divide only a few times before differentiating. They are sometimes called transit amplifying cells since they usually divide while migrating away from the stem cell niche. Both unipotent stem cells and progenitor cells have been called lineage restricted cells, but the stem cells have the capacity for self-renewal while the progenitor cells do not. Progenitor cells are usually more differentiated than stem cells and have been committed to become a particular type of cell. In many instances, stem cell division generates progeny that become progenitor cells, as is seen in the formation of the blood cells, sperm cells, and the nerve cells. First, we will be learning about adult stem cells. Adult stem cells. Numerous adult organs contain committed stem cells that can give rise to a limited set of cells and tissue types. In addition to the well-known hematopoietic stem cells, developmental biologists have discovered epidermal stem cells, neural stem cells, hair stem cells, melanocyte stem cells, muscle stem cells, tooth stem cells, gut stem cells, and germline stem cells. Such cells are not as easy to use as pluripotent embryonic stem cells. They are difficult to isolate since they often represent fewer than 1 out of every 1,000 cells in an organ. In addition, they appear to have a relatively low rate of cell division and do not proliferate rapidly. However, neither of these facts preclude their usefulness. Each year, some 40,000 bone marrow transplant procedures are performed in which hematopoietic stem cells are transferred from one person to another. These multipotential stem cells are rare, about 1 in every 15,000 bone marrow cells, but such transplantation treatment works well for people suffering from red blood cell deficiencies or leukemias. Techniques to selectively allow the growth and isolation of multipotent stem cells can allow some organ deficiencies to be treated in this way, as these blood cell deficiencies by administrating a source of committed stem cells in mice, very few, perhaps even one blood cell stem cell will reconstitute the mouse blood and immune system. A single mammary stem cell will generate an entire mammary gland, including epithelium, muscle, and stroma. And a single transplanted prostate stem cell will produce an entire prostate gland. Harvey and colleagues have shown that when neutral stem cells from the midbrain of adult rats are cultured in mixtures of paracrine factors, they will differentiate into dopaminergic neurons that can ameliorate and the rodent version of Parkinson's disease. In adult stem cell niches, many tissues and organs contain stem cells that undergo continual renewal. These include the mammalian epidermis, hair follicles, intestinal villi, blood cells, and sperm cells, as well as Drosophila intestine, sperm and egg cells. Such stem cells must maintain the long-term ability to divide, producing some daughter cells that are differentiated and other daughter cells that remain stem cells. The ability of a cell to become an adult stem cell is differentiated in large part by where it resides. The continuously proliferating stem cells are housed in compartments called stem cell niches, sometimes called regulatory microenvironments. These are particular places in the embryo that allow controlled proliferation of these stem cells within the niche and the controlled differentiation of the cell progeny that leave the niche. Stem cell niches regulate stem cell proliferation and differentiation, usually by paracrine factors that are produced by the niche cells. These factors retain the cells in an uncommitted state. Once the cell leaves the niche, the paracrine factors cannot reach them and the cells begin to differentiate. Mouse incisors, for instance, differ from human incisors in that they continue to grow throughout the lifetime of an animal. Each mouse incisor has two stem cell niches. One is the inside facing into the mouth and one is the outside facing the lips. The stem cells that reside therein are kept in a proliferative and non-differentiated state by an integrated network of paracrine factors including FGF3 and activin, which increase the proliferation of stem cells, and their respective inhibitors BMP4 and follistatin. Teeth in humans and most other mammals lack stem cell niches and thus do not regenerate. Similarly, mammalian hair follicles contain a bulge that houses the melanocyte stem cells which provide pigment to the hair. The division of the melanocyte stem cells within this niche is coordinated with hair growth. Moreover, upon cell division, one daughter cell remains in the stem cell niche, retaining its stem cell properties, while the other daughter cell migrates towards the developing hair shaft. The migrating cell is a committed melanocyte progenitor cell and it will reside in the matrix at the bottom of the hair shaft, producing pigment forming melanocytes. The mammalian hematopoietic niche is found in the hollow cavities of trabecular bones such as sternum, where the bone marrow resides. The stem cells are in close proximity to the bone cells or osteocytes and the endothelial cells that line the blood vessels. A complex cocktail of paracrine factors including WNTs, angiopoietin, and stem cell factor combine cell surface signals from Notch and integrin to regulate stem cell proliferation and differentiation. So hormonal signals and pressures from the blood vessel as well as neurotransmitters from adjacent axons also help regulate hematopoiesis. In many instances, germ cells are continuously produced in stem cell niches. In Drosophila tests, the stem cells form sperm reside in a regulatory microenvironment called the hub. The hub consists of about a dozen somatic testis cells that are surrounded by 5 to 9 sperm germ stem cells. The division of the sperm stem cell is asymmetric, always producing one cell that remains attached to the hub and one unattached cell. The daughter cell attachment to the hub is maintained as a stem cell, while the cell that is not touched to the hub becomes a gonioblast, a committed cell that will divide to become the precursor of the sperm cell. The somatic cells of the hub create this asymmetric proliferation by secreting the paracrine factor and paired onto the cells attached to them. Unpaired protein activates the JAK-STAT pathway in the adjacent germ stem cells to specify their self-renewal. Those cells that are distant from the paracrine factor cannot receive this signal, so they begin their differentiation into the stem cell lineage. So physically, this asymmetric division involves the interactions between the sperm stem cells and the somatic cells. In the division of the stem cell, one centrosome remains attached to the cortex at the contact site between the stem cell and the somatic cell. The other centrosome moves to the outside, thus establishing a mitochondrial spindle that will produce one daughter cell attached to the hub and the other daughter cell away from it. The stem cell niche is a critical component of our phenotype, and it does nothing less than regulate the ratio of cell division to cell differentiation. This means that maintenance of such niches is crucial for our health. Too much stem cell differentiation depletes the stem cells and promotes the phenotypes of aging or decay. Too much stem cell division can cause cancers to rise. The graying of mammalian hair in mice as well as human can involve a breakdown of the maintenance of the stem cell niche such that both daughters of the dividing stem cells differentiate, thereby leaving a smaller population of stem cells that cannot continue to make pigment-forming melanocytes. Melanoproliferative disease, a cancer of blood stem cell, and non-lymphocytic redivitis results when the stem cell niche is unable to provide the signals needed for proper blood cell differentiation. Thus, stem cell niches provide microenvironments that regulate stem cell renewal, survival, and differentiation. They are paracrine factors, cell adhesion molecules, and architecture allow asymmetric cell division such that a stem cell divides in a manner that allows one of its daughter cells to have a higher probability of leaving the niche and beginning to differentiate according to the new signals it encounters. Then next is about mesenchymal stem cells. Multipotent adult stem cells. Adult stem cells are used in the adult body to replace worn-out somatic cells on a regular basis. On epidermis, our intestinal epithelium and our blood cells are continuously being replaced with cells generated by dividing adult stem cells. Most, if not all, adult stem cells are restricted to forming only a few cell types. And hematopoietic stem cells marked with green fluorescent protein were placed in mice. Their labeled descendants were found throughout the animal's blood but not in any other tissue. Some adult stem cells, however, appear to have a surprisingly large degree of plasticity. These multipotent stem cells are called mesenchymal stem cells or MSC, sometimes called bone marrow derived stem cells, and their potency remains a controversial subject. So, originally found in bone marrow, multipotent MSCs have also been found in adult tissues such as fat, muscle, thymus, and dental pulp, as well as in the umbilical cord. Indeed, the first finding that human umbilical cords are deciduous, teeth contains MSCs, can lead some physicians to propose that parents freeze cells from their child's umbilical cord or teeth so that these cells will be available for transplantation later in life. However, the crucial test of a pluripotency, the ability of a mouse stem cells to generate cells of all germ cells when inserted into a blastocyst, had not yet been achieved. So, mesenchymal stem cells are able to give rise to numerous bone, cartilage, muscles, and fat lineages. The differentiation of MSCs is predicted on both the paracrine factors and cell matrix molecules in the stem cell niche. Certain cell matrix components, especially laminin, have been implicated in keeping MSCs in a state of undifferentiated stemness. So, certain paracrine factors appear to direct development into specific lineages. In one study, platelet-derived growth factor was critical for chordogenesis and fat formation. TGF-beta signaling was important for chordogenesis, and FGF signaling has crucial for differentiation of MSCs into bone marrow cells. In addition to paracrine factors, the repertoire of cell types from mesenchymal stem cells may also be enhanced by the surfaces on which the stem cells reside. Human mesenchymal stem cell differentiation. Human MSCs can differentiate according to the elasticity of the surface on which they are placed. If placed on soft matrices of collagen-coated polyacrylamide, these stem cells differentiate into neurons. A moderately elastic matrix of these materials cause the same stem cells to become muscle cells, while harder matrices cause the MSCs to produce bone cells. It is not known what this range of potency is found normally in the body. So, mesenchymal stem cells have recently been linked to normal growth and repair conditions in the human body. Indeed, one premature aging syndrome, Hutchinson Gilford progeria, appears to be caused by the inability of MSCs to differentiate into certain cell types, such as fat cells. These findings lead to speculation that the loss of MSC's ability to differentiate may be a component of the normal aging syndrome. Moreover, MSCs may work in ways other than differentiating into needed cell types. They may produce paracrine factors that aid other more specific stem cells to divide and repair tissues. New perspective, the ability to create, isolate, and manipulate stem cells offers a visible vision of regenerative medicine where a patient can have his or her diseased organs regrown and replaced by one's own stem cells. But beyond the potential medical uses, stem cells tell us a great many facts about how body is constructed and how it maintains its structure. Organs often form by the regulation of stem cells, and we will see that the skin, hair, blood, and parts of the nervous system routinely use stem cells in their construction. Stem cells certainly give credence to the view that development never ends and offer fascinating, if not frightening, potential ways to modify development.